Welcome, everyone. Thanks so much for joining us today, and I'm going to welcome our panelists, too, for joining us for a conversation about genome editing and the human germline. As many of you know, this conversation is for the Broad community, and the idea here is to really just sort of have a dialogue about what happened, try to understand what happened, but also jumpstart some conversations about what this means both for our community and more broadly for the scientific community as well as the world. And so to that end, we're lucky that some of the world's leading experts on both clinical genetics and genome editing happen to also be the Broad's experts on those subjects. So I want to welcome David and Heidi and Feng, and thank you for being here to have this conversation. Recent events at the end of November in Shenzhen and Hong Kong are really why we're here and why this controversy has begun. And so I want to ask David to just give us a sort of brief synopsis of what we know about what happened.
Right, so this is actually challenging to do in part because I want to summarize what was claimed to be done without, at least initially, rendering any kind of judgment. So I'll try to make this like the Planet Fitness ads. This is a judgment-free zone, at least for the first 3 minutes. But the target of the genome edit is a protein called CCR5. This is a protein that is known to be a co-receptor for viruses, including HIV. There are humans walking this earth that naturally have a variant of CCR5 called CCR5 delta 32. This is an allele of a variant of CCR5 that is missing 32 base pairs early enough in the protein, in exon 3, that the protein is rendered non-functional. And in humans that have 2 copies of CCR5 delta 32, which is maybe 1% of certain European clades, those humans are thought to be quite resistant to HIV infection. It's important to note that CCR5 delta 32 is not thought to be a universally beneficial mutation. First, it hasn't been, I think, that extensively studied as to its clinical consequences, but there are a couple papers that have been published suggesting that individuals who are CCR5 delta 32 may have increased risk for complications from West Nile virus and from influenza, among other diseases. There is a patient, a very famous HIV patient, Timothy Ray Brown, who had both leukemia and HIV, and his doctor creatively gave him a bone marrow transplant from a CCR5 delta 32 donor as part of his leukemia treatment. And he remains, I think, the only patient that the medical community has agreed has been cured of HIV infection. It's undetectable without medication for a long time. So Dr. Zhang He, a professor in China, decided to focus on genome editing CCR5 to try to recreate this CCR5 delta 32 variant. Now, he used the CRISPR-Cas9 nuclease tool together with a guide RNA that cuts near, although not exactly, where the CCR5 delta 32 deletion starts. As many of you may know, when you make a double-stranded cut in DNA in many eukaryotic cells, the cell responds by inserting and deleting nucleotides to create typically a mixture of variants. So it's not correct to say that he used the Cas9 nuclease to exactly recreate the CCR5 delta 32 variant. In fact, he created a mixture of variants which have insertions and deletions around the site that he targeted in exon 3. He did these editing experiments first in mammalian cells, in mouse embryos, in macaque embryos, and eventually in human embryos. He characterized the nature of the products he got out. One of the aspects of his data, as presented in his slides, that I was struck with is the fact that in his animal embryos, mouse embryo and macaque embryo studies, many of the vast majority of the products that came out were so-called mosaic embryos, meaning embryos in which the cells, as the embryo developed, represented a mixture of different genotypes. They had different variants of inserted and deleted CCR5 genes together with wild-type genes. And that mosaicism was quite strong in the animal embryos that he edited. Then he started working on human embryos, and he enrolled volunteers that were all HIV-positive fathers and HIV-negative mothers. He took the HIV-positive father's sperm, subjected it to a procedure called sperm washing, which you can probably imagine what that looks like, that is designed to remove the HIV virus from the sperm, and then in vitro used it to fertilize the HIV-negative mother's eggs, and then did experiments introducing CRISPR-Cas9 and the same guide RNA he had used in his cell studies and in his animal studies into those human embryos, sequenced the DNA from the embryos as they developed, and confirmed that he was getting the creation of these insertions and deletions at the CCR5, in the CCR5 gene, around the site where that delta 32 variant normally begins its deletion, although, again, you generate a mixture of different products. Some of those embryos included an embryo that was thought to have a 1 base insertion on one copy of the CCR5 gene and a 4 base deletion on the other copy. That we'll call embryo number 1. Embryo number 2, human embryo number 2, was thought to have a wild-type copy of CCR5 and a 15 base deletion in the other copy of CCR5. That 15 base deletion, of course, being a multiple of 3, means that that corresponding protein has about a 5 amino acid deletion, and as it turns out, because the deletion was perfectly in frame, no other mutations on the edges of the CCR5 protein. He recognized that there was ambiguity about the consequence of having this mixture of products that putatively could be used to initiate a human pregnancy. So he asked the parents whether the parents wanted to proceed with the pregnancy, noting that the heterozygosity of embryo number 2 and the 5 amino acid deletion introduced uncertainties about the function of CCR5 and what the consequence would be for the resulting baby. The parents elected to go forward with the pregnancy, and this resulted in the 2 babies from embryo number 1 and embryo number 2 with edited CCR5 genes. Now, that's the claim. The DNA sequencing results that Dr. He presented during his talk of the embryo and of the babies are not fully consistent with these claims, and that's something that,
Okay.
that perhaps we can
I'm
talk
going to
about.
cut you off there, so thank you. So assuming these claims are true, we have the first germline edited embryos that resulted in successful pregnancies, at least 2 pregnancies. So I want to ask Feng to weigh in here on what are the scientific concerns about how this was done and how this was presented from your point of view that are what's raised by Dr. He's experiment?
So just to elaborate on what has been done by Dr. He, the techniques that he employed have really been around for quite a while now.
Is your mic on?
I might need to switch the...
Oh, sorry. Okay, so just to elaborate on the technique that Dr. He used, the same techniques that he applied to human embryo has been around for quite a while. Scientists have been using exactly the same procedure to engineer either mouse embryo or primate embryo or many other mammalian embryos. The way it works is you just take an embryo and you inject the gene editing material into these embryos. So the technique has been around, and based on what he has described, he has performed exactly the same procedure. Now, the level of characterization that he performed on these embryos seems to be, on the surface level, seems to be quite comprehensive. He used a number of different techniques and sequencing-based validation at multiple points in multiple types of samples, whether it's tissue or blood or different types of maternal fluid to analyze mutation. But based on examination of the data that he presented, the quality of those data seems to be quite lacking in terms of the level of rigor that was performed to interpret those data. So based on what the analysis shows, I was shocked that he went ahead and implanted those embryos, because if the goal was to remove CCR5 from those individuals, then the result clearly shows that he is not able to achieve that effect.
So there's a fair chance that we've initiated germline genome editing without conferring the intended medical benefit of that editing. So that's a concern.
Right.
Yeah.
Heidi, can you weigh in a little bit about the medical concerns that you might have for Lulu and Nana, these two babies that were born with the deletion of CCR5?
Sure. So they released a report onto the website that is sort of the outcome of the study that is a ClinVar report, where basically they took the genome sequencing of the two embryos. As far as I can gather, there were no methods provided, and then ran it against the ClinVar database, which is one of the most commonly used clinical variant databases, and then created a report for each of the embryos designating variants that were inherited, shown in the parents, and those variants that were de novo, and then providing the interpretations that came from whoever submitted those variants to ClinVar. So if you read both of those reports, they didn't do any quality check for anything. So every variant in that report that says it's likely pathogenic or pathogenic is actually incorrect. And in fact, if you go into those variants in ClinVar, you'll see that 10 clinical labs say it's benign, and one rogue submitter that is actually excluded in ClinVar says it's pathogenic. So just some basic quality assessment. And in fact, I think nearly all of the variants exist at a population frequency above 25% of the population. So they're not pathogenic variants for rare diseases as listed in the report. In addition, they're actually probably unlikely to be de novo as well, based on what the alleles are. So that probably suggests that the quality of the parental sequencing that shows, in theory, absence of those variants maybe wasn't so good, and so they're assuming de novo when they may not be. The other, so that's got all sorts of problems in terms of that report. We have no idea what was actually delivered to the family. Was this Excel spreadsheet delivered in some clinical report? So I can't really say that this was a report given to the family per se, but certainly there's issues with that report itself. And also, one of the other reasons why this might have been done is to think about off-target impacts to the embryos based on the enzyme, potentially damaging other things in the genome, which is why you might want to look at de novo variation, right, that isn't in the parents. Again, have they looked at all types of potential off-target effects that could be chromosome abnormalities or large copy number changes that might be induced by double-stranded breaks that occur in the technology, and so this would not capture any of those. So I think, understanding that, I don't think any of us can really know what the true medical implications of that experiment from the limited information that's been provided. I don't know if either of you.
Maybe I'll make a comment about the off-target analysis. Just as Feng said, Dr. Hood did an analysis that sort of checks off the boxes superficially that you'd like to see with an off-target analysis. He used computational methods which have modest predictive value, and he used an experimental unbiased approach called digenome-seq. But really, his use of both of those types of techniques was, if you look at the data presented in the slides, was pretty superficial. The depth of his analysis and sequencing was also superficial. And I was left, now that we're out of the judgment-free zone, I was left with the sense that at just about every turn, there was a sort of superficiality with the scientific analysis that led to these rather weighty decisions. He showed the structure of CCR5 with the 5 amino acids that were deleted in this second girl, and said, well, I looked at the structure and decided that that would probably mess up the function of CCR5. But that falls so far short of saying, this is a protein you should put into a human that's going to then have this defective copy of the protein for the rest of their lives and pass it down to their progeny. it's just amazing. I mean, deleting 5 amino acids from a protein could create an aggregation-prone prion. We don't know. So the level of investigation was superficial. And I think that's also representative of the fact that there wasn't really an unmet medical need here. Because the HIV-positive father's sperm was already washed free of HIV. So these babies would have been perfectly HIV-free, no genome editing required, which then raises the question of why he actually did the editing to begin
So I
with.
see everyone nodding, so we're all kind of agreeing that this was not a great ideal test case of genome germline editing, to put it mildly. Is there an ideal test case? What if this were to go forward, what would you have wanted to see? Heidi, maybe you can start.
Yeah, I mean, you'd certainly want to see a disorder that no one would object to the grave consequences. Take a disease like Tay-Sachs disease, where a child is born apparently normal, and then when the first 5 years of life rapidly, degenerates in function and leading to death. No one probably would question a disease like that being a very serious medical consequence that families, would have a right to try to avoid. So, really wanting to tackle something, I have to balance those diseases that are amenable to the technology, but also trying to pick a target that really, without question, society would have a better agreement with, I think.
Well, this raises an interesting question because this work was not done with a sort of societal sanction. We're not sure how the Chinese government, it seems to me from reports that we're not sure whether the Chinese government was aware of this research, supported it, or whether they did not. But in terms of a broad societal, either among the scientific community or more broadly in society, there wasn't a sort of sanction for this work to go forward. I'm just to kind of push on that issue a little bit, is there a world in which we advance somatic gene editing and we, obviously we have the techniques already of embryo selection and IVF, where we would actually need to or want to use germline genome editing for even a Tay-Sachs more extreme case? Or are these cases always going to be about enhancement and protecting against a potential outcome as opposed to actually treating a medical need? Feng, do you have thoughts about that?
Yeah, I think that's a really good question. So it's unclear to me that germline editing for the application of treating disease is really all that necessary at all, because for most diseases, so the clear case where if a couple really want to conceive their own children, and genome editing in the embryo level is required, is if both parents are homozygous for a disease-causing mutation. And only in that case would the fertilized embryo with 100% certainty have a disease-causing mutation. The chances that happening, I don't know the statistics, but I think it's very, 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 very minimal. Outside of that scenario, there is always the possibility that you can conduct pre-implantation genetic testing to then identify an embryo that doesn't carry the disease-causing mutation. And many mutations which are recessive, as long as you have one copy that's wild type, then the person will not be affected by that mutation. And because of that, I think really most of what we're talking about is changing genetics for enhancement applications. And that's something that, because biology is so complex and we can't predict what would happen, it's something that we really cannot do right now.
Can I just
David.
add that I think those are good points, but there's quite a lot of gray area between disease prevention and disease treatment. Almost everybody in this room is homozygous for alanine 673 in your amyloid precursor protein. That gives you a much higher chance of Alzheimer's disease than the 0.1% of Icelandic people that have a threonine at that allele. So would you say that all of us, almost all of us, we're homozygous for the disease allele, and therefore if we procreate with similar homozygotes, our kids are very likely to also carry that much higher chance of Alzheimer's disease. So what's higher and lower also depends on what you consider your baseline to be. And it seems that we shouldn't only consider the most common risk as the baseline, because then that would lead to a weird argument that we should treat diseases on the basis of how frequent the allele is, not just how severe the disease is or what the unmet medical need of that disease is. So there is a lot of gray area in between where I think some would consider it enhancement, some would consider it disease prevention, some would consider it treating a serious unmet medical need like Alzheimer's disease. So we will increasingly face these challenges where opportunities will arise as these tools get more and more capable and more widespread, where it's not so clear-cut. I think, ironically, this case is actually, Dr. He's case is fairly clear-cut in the sort of no unmet medical need. But there will be, it's easy to anticipate a future very soon, maybe now, where there's lots and lots of gray areas, and the applications are, in some doctors and scientists' minds, are justified in going forward, and in others it wouldn't be, which highlights the importance of societal engagement and engaging the ethicists, the regulators, other scientists, which largely wasn't done in this case.
And I want to return to that in just 1 second. There's a sort of other end of the spectrum view. Carl Zimmer has written in the New York Times that this is actually, his view is that this is actually not such a big deal, that with donor mitochondria from egg donors, that there's already been a sense of germline genomic modification. And I just want to ask you all to say, well, in what sense is this a big deal? Or are we just sort of at the bleeding edge? Obviously, we've decided this is not a good test case. But to your point, David, are we sort of just at the edge of a new frontier in which this is going to become more acceptable?
So one thing to think about is, as Feng pointed out, there are plenty of scenarios where this is actually happening without genome editing in terms of IVF, so you can select embryos to implant. So in essence, we are manipulating the outcomes of downstream pregnancies by selecting against embryos that have certain defects. And so from a societal perspective, I actually don't see a huge distinction between editing an embryo to lead to a better outcome versus selecting out an embryo that has a defect. They have the same outcome, right? Now, there have been some controversies that have arisen in the past because we can do this. So, for instance, one of the most common congenital defects is hearing loss, and over half of it is genetic. And you have a very large, well, not very large, but a segment of society of deaf individuals that are culturally deaf. They don't believe they have a defect. They are proud of their deafness, and they would like their children to be deaf too. So there have been ethical circumstances in which those families have selected for, through genetics, a deaf child. There's been at least one example of that. And so that brings up a societal question. Should an embryo without any choice be selected for a defect? It's just a different scenario around the same societal question. So I don't think this is a new topic. I think it is continuing a conversation about what we think should be individual decisions versus societal decisions.
So that's a great segue. I want to kind of turn us to where we should go from here. Sort of what should be the response of the scientific community? What should be the model for engagement going forward? So there was something of a call for a moratorium from the 2015 Gene Editing Summit on this, on germline editing, and a call for a broad societal consensus. And we saw a statement come out of the 2018 summit that seemed to soften that. And David and Feng, you were there, so maybe you can explain to us, if you can, why that was kind of softened to this idea of attention to societal effects. Reflecting on that, what should be the standard by which this goes forward? Should there be a ban? Should there be some sort of process for getting societal buy-in to this? How would that work?
So I just want to add one point. So in the past week or so, I've heard people compare the experiment that Dr. He did in editing human embryo with IVF and also with the mitochondrial transplantation. And the argument has been made where people are arguing that gene editing is an important advance and is just another one of these new technological breakthroughs that continues the evolution of fertility techniques to allow parents to conceive children that sort of continues from IVF and mitochondrial transplantation. I think that our argument is flawed because if you look at the case of IVF and you also look at the case of mitochondrial transplantation, there are clear medical need and clear benefit that can stem out of these different techniques. In the case of gene editing, there was no clear medical need and there's no clear advantage to performing this technique in the case that Dr. He performed. And so I think in thinking about how we go and practice these techniques and how we justify the applications of techniques, it's really important to think about what is the potential advantage and what is the potential risk. And there really needs to be a very clear potential advantage, clear benefit to the people who are going to be affected by this before we move forward with any of it.
So who should make that decision? Should it be the father who wants to see his daughters born? Should it be the scientists doing the genome editing?
I think there are certain, so first to go back to your earlier question, I think it would be very wise to agree to put a moratorium on creating human pregnancies from edited embryos at this time. I don't think this is a moment where it's good for scientists to be nuanced and equivocate about certain principles. This is a time where, even if there were a much more serious unmet medical need, a lot better science has to be done than what was done in Dr. He's case, in my opinion, before we create babies from these edited embryos. So I feel strongly about that point. And so to answer your earlier point, it was a little bit disappointing to see that the statement that was immediately issued by the summit organizers following the Hong Kong summit. Of course, they were under extreme time pressure, and I understand the furor and everything, but it seems to be, as you pointed out, a softening, not a strengthening of position with respect to what are the circumstances under which germline, that is embryo editing, would be acceptable. I think
maybe
we
an
can,
unintentional softening is what you're saying.
I don't know if it was intentional or not.
Right.
I wouldn't try to second guess their motivation, but I think it is it's a wake-up call and a moment that we can use to try to define much more crisply what are the factors that weigh positively and negatively into doing germline embryo editing, and of course, ultimately in taking some of those edited embryos and thinking about turning them into babies, which currently I have a really hard time seeing the circumstance under which you'd want to do that.
Okay, we're going to turn to audience questions soon, and I'm going to put up the number. If you would rather text your question than come to a microphone, you're welcome to do that. And while people are sort of formulating their questions, I'll just ask the question of how exactly, you're calling for a moratorium, so how exactly would that moratorium be enforced? Are we always going to see Dr. He's and parents desperate for an enhancement or what might be a perceived medical need, making decisions like this? How would that work?
I think there's many layers that help regulate the scientific community. There's, of course, a funding layer. There's the government. We generally assume we don't want to break the law layer. And then there's a huge community peer review layer. And all of those have collectively, historically, contributed to minimizing rogue scientist activities. Ultimately, I think we're a little bit at the mercy of each other's reasonableness. And if somebody really wanted to do a rogue experiment, just as if somebody really wanted to do a rogue action in society that has nothing to do with science, it's pretty hard to stop them from doing that. But I think we can, do better and strengthen the layers that we already have, and make them very clear with respect to this issue. Incidentally, there's a big movement now to think about the relationship of peer review and science publication, and I encourage people to think about the lessons from this fiasco as they relate to whether it would be better for scientists to publish more of their research without peer review. Given that failure to engage the scientific community, none of these results have been peer reviewed yet that caused this furor, is one of the, I think, indictments of what was done.
Thanks so much. Okay, and we also have questions that have come in from online, and I think someone may be sharing those with us as well, but it looks like we have a question here in the room. Thanks.
I actually have two questions. The first is about the institution where this took place. You mentioned that the Chinese government may not have been aware of what was happening, but was there an institutional review? And what's the reputation of that institution? And my second question was about consent, because I thought I read that there had been some sort of consent, but it was unclear if the parents thought they were just consenting to the IVF treatment or if they actually understood what was planned.
Those are great questions.
Go ahead. No, go ahead.
So going back to the question about moratorium, I think there needs to be legislation and there needs to be laws sort of regulating whether or not this can happen and whether or not it's legal to do this. So
Internationally?
Well, we need to start one step at a time. So if we can start with one country, 2 countries, 3 countries, and so forth, that's a good place to begin. Internationally, I think it would be hard to do that across all of the countries at the same time, but we need to start somewhere. And the US is not a bad place to start. China is also not a bad place to start. Going back to the question then about what happened in China, it's unlikely that the Chinese government didn't know about this. So the summit was supposed to be held in Beijing. And in February, the summit, somehow the Chinese Academy of Science pulled out of the organization of the summit, and the summit was then relocated to Hong Kong. For some reason, they couldn't find an auditorium to fit 500 people in Beijing. If you think about the timing works out well. Well, I mean, not worked out well, but the timing fits because that's probably when maybe the experiment began or when the implantation happened and was found to be successful. The response by the Chinese Communist Party mouthpiece, the People's Daily, immediately following the break of this news was that this is a moment of national pride. We made a seminal breakthrough. And only after this sudden international outcry condemning this work that they replaced that article to say that we're beginning an investigation. We had no idea this was happening. The institution that Dr. He belongs to is a new university in Shenzhen, south of China. It's a university that's about 10 years old. And it's a university that's, they're trying to make a name for themselves to establish a strong reputation internationally. And so the university went out and publicly say that they had no awareness of the work that was happening and that Dr. He had been on leave since February this year. That timing also seems quite strange.
Count the months.
And they actually had the AP for weeks, if not months, ahead of the story, filming in his lab because they had coordinated the release of this information. So it's not actually correct that the story was, quote, leaked to the press. There was actually a reach out, presumably with the institution's knowledge, although the institution and the government, of course, now disavows any knowledge. But they engaged the Associated Press. The only reason the news broke when it did was because MIT's Tech Review started to scoop the Associated Press.
Do you want to say a word about IRBs and
so let me just finish. So the way that the Chinese government reacts to this and how the government sort of treats this example of gross violation of the ethics and also sort of complete destruction of public trust for scientific community is really important. If there isn't a strong signal, who is to say that there won't be 100 other similar type of experiments that will take place, not only in China, but in other medical tourism hotspots where people are going to go ahead and try this. And so I think strong regulation, beginning somewhere, is really important. And then moral leaders or ethical leaders or countries that have leadership in ethics and morality, hopefully will be able to lead other countries to then follow suit and then begin to implement a moratorium.
Yeah.
It's a great question. Who has moral authority in the world today?
I just want to jump in because moratorium make a law. There's things to think about practically. I'm right now dealing with a law in Massachusetts that prevents any doctor who didn't order the original genetic test from getting the results. Why is that? Because many, many years ago, when Huntington's disease was the only thing we could test genetically, it was felt there was so much stigma that no one should see a patient's result except the ordering physician. Well, this has created a huge problem in use of genetics in healthcare in this state. But we are beholden to lawmakers to get things changed, which I'd rather not be beholden to politicians and their agendas to actually rapidly move forward with science. So I would rather see a thoughtful group of scientists with the appropriate ethical and other societal participation, develop guidance for IRBs and the many other aspects that control science and clinical trials in our society, be the ones that can iterate on this as opposed to laws. Just a very small point.
A fair point, and we might return to that later in sort of how scientists build that trust to be able to do that in terms of the public trust. Could someone just say a word about how the consent was done, and then we'll turn to this question, because I don't think that part of your question.
It's complicated. The consent forms have been published online. They've been criticized. Dr. He apparently, and his postdoc, I think, consented the patients themselves, which is generally frowned upon, especially if your doctor leading the study is a physicist by training. And so there is a real question of whether the consent was done to a reasonable standard that the general medical community would accept.
And also some ethical issues raised by the fact in China, HIV-positive patients are prohibited, I believe, by law from doing IVF. So there might have been some issues around equity and taking advantage of a situation of desperation on the part of the parents. This question over here, and then if there are any questions from online to read, feel free to chime in.
Thank you. And I think a lot of discussion on the benefits and the risk and necessity of this whole event. I'm interested in, so what's your idea if we know that the genome editing for the embryo for some specific case, the risk is clear and the benefit is clear and the necessity is good. So what is the boundary of this kind of research and the clinical application? So if we know that, another related question, who will decide whether this is necessary? Maybe the father or the parents think it's necessary. And then is that good or right to allow this kind of thing happen? Where is the boundary of for research or the application?
So the question of the boundary between research and application, and then who gets to decide when this is a need?
Those questions are not unique to genome editing, and just about every new drug, every new medical technology that's been deployed has had to answer those questions at one point or another. So there are processes involved that involve assembling a lot of preclinical data and clinical data, engaging with regulators, ethicists, the FDA. So I'm at least comforted by the fact that there are historical examples that have led to the effective deployment of medical techniques and new therapeutics, even in cases where early on the risk-benefit was less known or even was more dangerous than the more refined versions as they went through clinical use. So it's easy to predict that, for me, that I'm comforted by the fact that I imagine such a future will eventually happen for genome editing as well. But all of that required the full engagement of the scientific community, of regulators, of ethicists. And obviously, there was a failure to do so here. I think we all don't want, which really echoes Heidi's comment, we all don't want the laws, the regulations, the standards to change in a way that delays or prevents genome editing therapies in the future from actually reaching the patients that stand to really benefit from them. We just have to do so in a way that minimizes risk to patients and minimizes additional disasters that will only serve, ironically, to further the timeline between when the patients can get these treatments. So we have to proceed, but proceed really carefully. And if we don't take a strong enough stance condemning the missteps and working to make sure they don't happen again, then I worry, ironically, we'll just end up preventing patients from getting the treatments that could actually benefit them and be very safe and very efficacious.
I think this treatment is different from the other treatment because others only have the risk or benefit for the individual, but this one really added the genetics that will pass. Maybe for this generation there is nothing, but when it passes to others, combined through marriage or something, and
Sure.
will give some additional risk to
And
other
the distinction you
gens.
made in
So
your statement,
it cannot,
David,
it's
right?
not just affect your own kids, but also maybe affect all the other kids.
Yeah, that's right.
And the human genetic pool, which I
Right.
think you
Yeah,
raised
yeah.
in your statement, David, that this is a question about future generations. I think that's a very good
Yeah.
point
Yeah, that's
to
right.
raise.
And this is why germline editing, in contrast with somatic cell editing, is something completely different, because it cannot be applied in a focused, patient-derived, pre-consented way, where the consent is given by the patient and all the patient's progeny. That's not possible with germline editing. So I think your point is very well taken. I think it's the case that even non-germline genome editing medical treatments do affect future progeny, but not in a way that's as fundamental and directly controllable as germline genome editing.
And not just future progeny, but really the future of society, because we're talking about future generations.
Sure.
And Todd.
So I just wanted to follow up on this last discussion, maybe in the context of medical intervention generally, where the extent of the medical need matters, but it doesn't matter that much, actually. The bar is very high with respect to safety. It's a highly regulated process. If you can't give snake oil to a patient just because they have a horrible disease. There's a high bar of characterization of that drug, which essentially doesn't affect future generations at all. You just can't go around doing that sort of thing without a very high level of preclinical safety characterization. So it's easy to criticize this because the HIV indication wasn't medically indicated in the first place. But the notion that we would lower the bar for something that affects future generations rather than making it super high just seems totally crazy.
I mean, one practical thing to think about is today anyone can terminate a pregnancy for their own reason or no reason whatsoever, right? You can also get genetic testing on a lot of different things. So how does our society actually control decisions around prenatal testing, what can be tested for, and therefore what allows a couple to make a decision on a pregnancy, for example. So largely clinical labs today kind of control that, because if I get a sample in my lab and it's a prenatal specimen, we will decline to do certain tests, and many of the clinical labs do that. Some labs will not test for variants of uncertain significance. So there is some practical side to allowing some decision making. That said, most laboratories will accept DNA, extracted DNA specimens for testing, where the amniocentesis and the cells might have been grown up in another lab, extracted DNA, and then sent to the lab for testing. In that case, the laboratory often doesn't know what that specimen was from. And so a laboratory that's, for example, offering eye color, various traits, fast twitch, slow twitch, muscle that relates to endurance. A sample could just show up in a lab. It's extracted DNA. You have no idea where it came from. You run a test and you give a report back, having no idea that information could be used downstream for any sort of decision making, because we allow a couple to make a decision about a pregnancy, without judgment. So there is a notion that there is some degree of control we may not have. That said, since I work in this field, the vast majority of prenatal testing, the laboratories themselves control what specimens they will allow testing for.
I think.
First, thank you to the three of you for doing this, and to Bina for moderating. a quick comment and then a slightly complex question. I think we may have lost the opportunity for self-regulation. I think it would be totally appropriate for there to be law that is actually hard to change, because it should be hard to change. We need to amass a serious case to say we should go forward. And that brings me to my question to you. You know, Feng said there's not a particularly compelling case with regard to these genetic diseases, because in Tay-Sachs, 3/4 of the embryos are going to not have Tay-Sachs. And almost always you will find an embryo to reimplant, and if you can't, you could do, you know, sperm donor or egg donor or adoption. So this is only about whether or not a couple has an absolute right to have a child who's genetically related to both parents. That's the thing we're talking about, and maybe there is. But David, you then swung all the way over to the rare allele in Iceland that conveys protection to Alzheimer's. So I raised the safety questions you guys to think about, because you all think about human genetics. There's the FUT2 allele, which our colleague George Church argued to me we should install in everybody because it protects us from noroviruses, these horrible things you get on cruise ships. Then I went back, well, it's 2 days, it's terrible. I had a norovirus once, terrible. But I went back to the MPG program and chatted with folks, and it turns out that same FUT2 knockout also substantially increases your risk of Crohn's disease, such that it would lead to about 100,000 new cases of Crohn's in the United States, which has a 10-fold increased risk of colon cancer, et cetera. Now, thank goodness we know this because of the MPG program, but we don't necessarily know that for common alleles. Then when we get to the rare allele like Iceland, how do you propose to define the safety of this allele that we should install in everybody to protect against Alzheimer's, including the genetic interactions that it will have with other clever things that are installed? And then what if we go beyond naturally occurring alleles to clever circuitry, that you put in a clever circuit in one person, a clever circuit in another person, and a whole generation of marriage counselors are going to have to arise to say whether this is an auspicious match between the new circuitry here and the new circuitry there. Mm-hmm. So how do we go from these seemingly heartbreaking cases of genetic disease where, in fact, we can already make sure there's no child born with the disease, that's not the issue, to common alleles, to rare alleles, to new circuitry? Are we going to have in place something that is robust enough that there's not a compelling case constantly being made for scientists for their latest clever idea?
That's a great question. I assume the correct answer is not we should start Broad Iceland. But you know, I think the point is very well taken, that as you get to alleles with frequencies less than several billion people and several million people and several thousand people, the uncertainty goes way, way up. And I think that should be one of the factors that the regulators and the FDA and scientists use when deciding what alleles to advance towards the clinic.
Yeah. And you know, there's a lot of other circumstances in medicine where you take the data that you have, you make recommendations, and then years later, like hormone replacement therapy, you know, a reversal of those recommendations. There's been, you know, even like whether you should eat butter or margarine. I mean, and you know, there's all sorts of things where, based on the science at the time, certain recommendations were made. Years later, those get reversed. We've introduced entire species into areas to eradicate certain disease, and then those species took over and caused a new problem.
But human medical recommendations wash out in a generation or two.
Yeah,
What's the recall notice that you're going to send out to the population to recall some of these things?
Well, it's also, though, hard to eradicate a gene in an entire population rapidly either, though, right? I mean, you know, likely, if somebody really thought it was a good idea to put this Icelandic rare variant into the population, it's going to take effort to disseminate it, right? So hopefully, within a certain amount of time, you realize, oh, there's downsides to that variant, and reverse it. I'm not saying we should do this, but.
Well, so,
speaking of marriage
so
counseling.
since, so I have something to add. So, well, just to play devil's advocate, since we are talking about science and technology, if genome editing works well, in the future, we can put in any mutation we want, and we can remove any mutation we want. So the issue that, can we recall something, I think is less of an issue because we'll be able to get rid of it because genome editing works really well. Anyway, that's only to play devil's advocate.
You're very confident that it's going to be equitably available.
It's just
to all the
Well,
population.
in the ideal case, we'll be able to do all that.
It's actually a good-
but,
there is
but
a-
actually, so the other thing is,
It's a phone app.
no,
We're
but,
gonna go
but
back
I think,
to the
I think
public trust
but
question.
I think that the more important thing to ask is, I think a really good test that I found to be helpful is, would I do it for myself? Would I do it for my own kids? And I cannot think of a scenario where I would say yes to the application of this technology. And I'm a pretty informed person. And when it comes to consent, it's not consent, it's informed consent.
Right.
And
right.
so the patient has to be informed.
Yeah.
And even for someone who is very informed, and I couldn't even come to agreement to say yes.
Right.
And I think that's a pretty clear.
And that means just for your own child, you wouldn't
Right.
do that, right?
Exactly.
Let alone the unknowns and uncertainty, the risks through the population.
I would think of not having children first before I would apply
Or
that.
not having biological children, or
Right.
fully biological children. One more question. A question from online. How about this one first, and then the one from online?
So I texted my question, so I'm sorry if that's the
Oh.
one you're going to read. I just, got a little bit impatient. But this is this is back to the public trust question. I think we can all think of examples in terms of the medical community and certain populations where for each technological advancement, there was a group or subpopulation that was really taken advantage of in order to sort of move technology forward for the betterment of society. So how, or I guess, what are the best practices in this case to sort of prevent, for example, a mad quote unquote scientist from saying, well, there are laws in the US, let me just go to X country and run my experiment. So how do you sort of see advancing this technology without really taking advantage of any one group?
Yeah, I mean, I guess I certainly feel there was a major failure in messaging in this sort of scenario, and that if we had actually had some sort of, how many years ago was it when the recombinant DNA technology was being introduced, and there was a thoughtful committee convened to really examine the potential downstream effects of that, assemble experts, and come up with recommendations, and then allow that technology to roll out in a thoughtful way based on the input of what was believed to be a fairly thoughtful process. So, I think while there has been certainly, and David, if Feng can weigh in better than I can on this field, I think there has been thought there, probably not enough to really think about all of the different scenarios and weigh in, and that we're getting buy-in before these things happen. Because clearly, as you describe, there was a perspective probably that this was a major breakthrough and this was awesome. But if there had been a conversation with the people who, right after it happened, thought not so much, then that could have been adjudicated before we got to the point we're at. So I think we do need to sit back and have some thoughtful conversation about what are the right experiments to do? What does our society deem as useful ways to use this technology? And if you read about some of the startup companies that are operating this space, that over the last week I was trying to educate myself on a lot of things in this field that I don't actually work in. I think there are some very thoughtful ways this technology being used, mostly in somatic, examples. But I think we really want to engage the community in how this technology can be used for the benefit of humankind, where no one would really debate it, and use that as our messaging platform, as opposed to crazy scientists did X, right?
I do think the discussion. I do think that scientists, we could have done more. Mm-hmm. So, for example, this particular case with CCR5 was never clearly articulated as unnecessary and unjustified. Mm-hmm. And if there was some discussion, some public, sort of highly visible document that stated that application of CCR5 knockout in a human embryo is absolutely unjustified from a medical perspective, the regulators would probably have seen that.
How would you have known what front someone would have tried first? I mean, you could have a specific statement on every single gene as it's developed, but that seems to me like trying to anticipate something before you know it's happening. And I think the broader statement made by the scientific community, well, they were anticipated in that people knew it was a target.
So there was a thick multi-100-page report produced by the Human Gene Editing Working Group.
Right.
And in there, the discussion about what constitutes medical necessity was very ambiguous, very vague.
Got it. So you're saying be more specificity to that, those kinds
and instead there's wide discussion that CCR5 is a cure for HIV.
Right.
And so if there wasn't clear articulation of what the pros and cons of this is, regulators in China, I'm sure they probably thought this sounds like a good idea, but had they been more informed, this may have been prevented.
So I think I want to close by asking you all a question. So this was generously provided by the communications team here at the Broad. It shows the spike in Google searches for CRISPR, one at the time of the 60 Minutes episode and this most recent one. So you can see the attention being given to this issue right now. And this is really a question about public trust and sort of the question raised by Eric's comment, which is that have we already breached the public trust? Is there a chance to restore that? When you look at this and think about the attention that in the last few weeks that this issue has been given, what do you hope the public will take away from it? What do you think that scientists need to do to ensure that the takeaways are not misperceptions?
Maybe I'll start a part of answer to that question by noting that recently I saw some data about what segments of society are most trusted by consumers, by the public. And it turns out that scientists who practice in a particular field were the most trusted of 10 or 12 categories, followed closely by PBS documentaries, and et cetera, et cetera. Time magazine was way down the list, not to pick on Time, I mean general mass magazines. And even doctors were not quite as high as I thought they would be. So that helped to highlight, for me, the importance that we all proactively engage with the public, with the media when asked, to offer our thoughts and to try to be as broad-minded, as informed through discussions like this as possible. And so I think a big part of the answer to your question is, yes, we certainly did not do ourselves any favor as the scientific community by what happened. I'm sure nobody in this room thinks that they were directly responsible or contributed directly to what happened, but we all own it. We all have some responsibility to try to maintain, restore, and maximize the public's trust in the science that we believe in. And a big step towards doing that is to represent our community of scientists as well as we can, to talk to the neighbor who really wants to engage, even who has a completely opposite view and who you think is totally misinformed. Those types of engagements work. I recently offered to my daughter's class, middle school class, to go there and talk to them about what happened. And that kind of engagement, I think, is an important step.
I'd also add, I think you can think about the parallels with genetically modified organisms, GMOs, and some missed opportunity and messaging that happened when those were rolled out, largely viewed from a societal perspective as Monsanto and other major, commercial entities controlling. And I think GMOs got a much more negative view because of the way that whole thing unraveled. And it has led to an entire market and distrust of anything that's GMO. And you now go to the grocery store and you see GMO-free, all this stuff because of the public's distrust of that whole segment. I think largely because the messaging came from companies, not scientists. And I agree with David that if we can really own this as scientists, because there is larger public trust there, I think it'll help us direct the messaging around this, highlighting the good and how we're thoughtfully trying to control negative downstream impacts.
Yeah, and just And I absolutely agree that I think scientists have a responsibility to explain what they're doing, and it's only by helping the public understand what we're doing that they can know and feel that they can trust us. At that same time, I think there needs to be more tools provided to scientists to help them explain what they're doing. It's not about messaging. Messaging sounds kind of conceited, actually. It's really explaining what we do. And so the more we can do to help scientists develop communication skills, ability to more sort of simply reduce a complex scientific phenomenon down to something that even my grandmother or grandfather can understand. I think that's what we need to get to. And I think that's something that scientific mentors need to do to help their trainees, and then something that trainees can work hard to develop.
Well, unfortunately, we're out of time. I feel like we could talk for hours about this. Thank you so much to our panelists for taking the time with us. Thanks to everyone.